So before I begin, really would like to thank Professor Nathalie to have agreed to come down here and share her rich experience of last 30 years in the space of phytochemicals. And you have also already come to know what leaps they are making now in the field. And in fact, I would like to mention that Vasant is her first PhD student. So you can imagine the legacy, how it is continuing. So
continuing to work together.
Yeah. So now he's a colleague. But you know, she has been instrumental in building the lab and to bring it to a level where now you can see what results are coming out. Now, and again, tomorrow we are going, although this was her last lecture, but tomorrow, as you know, we are going to have one Q&A tutorial session. That will be your opportunity in this particular course to clarify if you have any questions with her, to discuss with her, make use of her presence, and see how best you can take it forward in your future endeavors. What I'm going to talk about now will be more of an exploratory topic in this domain. I'll be talking about microbial endophytes. And there has been a lot of talk, if you'll see literature, on microbial endophytes, whether or not they can be sustainable producers of phytochemicals. So there is still a lot of question marks around this particular subject. Very, very interesting, because you will find the topic becomes all the more interesting if you find contradictory results being published. There are equal number of results where people have observed production of host plant associated phytochemicals in these organisms. There are similar number of reports where people have experimented, done some molecular techniques, and have said that they cannot be sustainable producers. So the work is going on both the sides. And that's the best way, I would say, wherein it becomes really something to be explored about. So how we came to this idea was around 2010 when I joined the institute. There was a paper which was published on a microbial fungal endophyte producing camptothecin, which was isolated from Nothapodites pneumoniana. This is a plant which is known to produce a very high value compound, which is called as camptothecin. So as I've deliberately called it as exploratory research, because this is still a topic which is being explored around in the world. And it is worth mentioning this also because whatever cues you can get through this research can also be adapted to even creating GRAS platforms for production of the host plant metabolites. Now, what are endophytes? Just to briefly, for those who do not know, an endophyte is an endosymbiont, often can be a bacteria, can be a fungi, that lives within a plant for at least some part of its life without causing any apparent disease. So there is a lot of discussion even around the role of endophytes in the plants. At present, there is quite an excitement around endophytes with respect to production of high value metabolites per se. Can be as biocontrol agents, can be as biofertilizers. So there's a lot of excitement around endophytes going on. Now, this is just a small example, although there are plethora if you'll start digging in literature. There are certain examples of very high molecules, phytochemicals, which have been known to be also found in the exinic cultures of these endophytes, which were isolated from the host plant. And why do these molecules are called host plant associated molecules? Because these endophytes were isolated from the plants which were known to produce these phytomolecules. Now, the first step is bioprospecting of these organisms. there is enough discussion going around. I still remember my first presentation. We got funded from one of the government funding agencies, and I went to defend that proposal. It was a funded project, and this was my second year of. I was a young PI, and I was defending the leads from the project. And there were enough people in the committee who were questioning on the project itself that no, it is not possible to even culture endophytes. So what I'm trying to tell you is that it's completely exploratory, wherein people do believe that endophytes may not be even cultured exonically. So what are the critical factors which should be kept in mind when we want to project endophytes as a sustainable producer, as a high yielding strain for the phytochemicals? It has an evolutionary relationship with the plant. The site of product synthesis and storage in the plant should be known. So that, because when you are bioprospecting endophytes with a hypothesis that the endophyte may also be a producer of the host plant associated metabolite, it's also logical to also know from which part you should be trying to isolate the endophyte. A high yielding explant. This is one of the observations. I'll talk about a case study from the lab where we were trying to also, what we observed was that some kind of a correlation can indeed be drawn between higher yielding explants vis-a-vis the endophytes isolated from them, which were producing higher yields of the host plant associated metabolites. Now, another important factor which we have also explored is higher tolerance towards toxic intermediates or the final products in these potential endophytes, which can be bioprospected for such phytomolecules. We have also observed in our study wherein there is a possibility worth investigating whether the potential endophytes which you want to discover producing these phytochemicals, they seem to have higher tolerance towards the toxic intermediates or the end product. Sustainability analysis with respect to product synthesis is a very critical factor. Now, one of the bottlenecks, which is a well-known bottleneck with endophytes, is that they seem to have shown inconsistent production of this phytomolecule in the exinic state. Most of these reports which are published in literature, even the table which I showed you, in the first or few initial subcultures, one is able to observe the presence of the phytomolecule in the exinic state, but subsequently the capability is lost in the subculture. So which means that they cannot be reliable producers of these phytochemicals. So that's one aspect which is also worth investigating. Now, I'll be talking about certain optimization strategies as well, where we generally use optimization strategies to enhance the production of a phytomolecule in a production platform. Can be yeast, can be plant cells. Now, similar strategies have been reported in these potential endophytes, wherein people have seen enhanced production of the host plant associated metabolite, wherein people have tried to optimize the media components like carbon and nitrogen source and have seen in quite a fold enhancement in the host plant associated metabolite. People have worked around with culture conditions, the same strategies which we have learned in the course. Okay. Elicitors, we studied in plant cells, as well as when Professor Nathalie was discussing about microbial platforms. Indeed, stress inducers can play a role in enhancing these secondary or rather specialized metabolites in the cells. Now, people have tried metal ions where to improve camptothecin and its derivative productions in some of these endophytes. People have used synergistic impact to see if that can be multiple fold enhancements. So the strategies, if you'll see that they remain the same, the implementation process can be different. Similarly, people have tried precursor addition. In this particular case, camptothecin production from enterophosphora was increased by the addition of its intermediates of the pathway. Now you know tryptamine tryptophan, all these, where do they lie? So they have reported, indeed, they have observed that if you apply elicitation strategy, if you apply precursor addition strategies, you may be able to observe enhancement. The key is that the production has to be sustainable. Which means the production platform, one of the important factor to be kept in mind is any industrial process, it is transferable, or you can do a tech transfer only when it is reliable, the production platform, which means it has to be a sustainable producer so that you can create a master cell bank. So the production should not keep varying with every batch. So that has been one of the major bottlenecks in exploiting endophytes further. So it is still exploratory right now. Now, people have even tried epigenetic regulators. Now, with the hypothesis that maybe what happens that the minute you isolate these endophytes from the nature, from these explants, when they come under exinic state, their biosynthetic machinery gets cryptic. Why so? Because they are not in planta now. Maybe because of the missing stimuli in planta, because of which the biosynthetic machinery gets cryptic, and due to which you see a loss in their biosynthetic capability. That's also one of the hypotheses. So with that hypothesis, to overcome that or to even verify that, people have tried using epigenetic regulators as exogenous additions in the spent medium or in the medium to see if the production can be revived. And in one or two cases which have been reported in literature, they indeed did find that by adding these epigenetic regulators, the enhancements or the productions have been revived. We know now that after integrating, when you are developing a bioprocess, if you can integrate one or more of these strategies together, it can lead to multiple fold enhancement in the phytomolecule. So similarly, in this case also, in production from trichoderma, this is again an endophyte which was isolated, and where they have tested it for camptothecin production, and they observed 50-fold enhancement by doing this integration of all these optimization strategies, which we now know for productivity enhancement. So all these become very promising results to keep exploring these organisms, to still find if there is a possibility to find if they can be an independent producers of host plant associated metabolites. People have tried co-cultivation of endophytes, and they have found that because now when the endophytes are residing in the plant, there is a lot of metabolic crosstalk which can happen. They also want to be present in the plant, so they are in constant metabolic crosstalk with the plant environment. They are also in crosstalk with the microbial environment which is in the plant, because there might be a competition between microbes to reside in the plant. So there could be some quorum sensing a role to play. So there is a lot of metabolic crosstalk which is happening. Now, how to exploit it in vitro is through co-cultivation. When you do not know exactly what is the metabolic crosstalk, one of the ways of not being rational, but one of the experimental ways to go about it is to do a co-cultivation and exploit that metabolic crosstalk, which might be acting as a stimuli to enhance the production of your desired molecule. So people have tried more than, so they have tried with Alternaria and Phomopsis, and they have found an increase in Taxol production. Similarly, for Bacillus with Fusarium has led to 78-fold enhancement in the yield. So there are enough reports, if you will see, where people have explored many of these tools and strategies which we can implement on various other platforms, including your microbial cells or plant cells, and they have seen to be working for that particular phytomolecule. Now, in one of the lectures, I think one of the teams was mentioning about the co-culture of the plant with a mycorrhiza, I suppose, as an elicitation strategy. So that we use as an elicitation strategy for plant cell suspensions. But the same holds true for the microbe as well. So with that hypothesis that there is a metabolic exchange which might be giving a stimuli to both, if really both the organisms can produce the same molecule, then how to exploit it? So people have even tried co-cultivating the endophyte with the host plant cells. But there is an important point to be noted. Plant cells, they grow slow. Microbial cells will grow faster. So there is always a chance that one will overgrow the other. So what people, and in the end, when you talk about metabolic crosstalk, it is mainly about the metabolites exchanges. So how to avoid? So there is a very good example in this particular paper where they have used they designed a reactor and they made a semi-permeable, selectively permeable membrane such that only the metabolites could be exchanged, while the cells on both the sides, they don't have to be in contact with each other. See, 38-fold enhancement in paclitaxel. Now, just to give you what we had done in this space since 2010, just to explore and as an exploratory project ongoing in the lab, was we knew that camptothecin is one of the key molecules as a lead molecule required for the anti-cancer drugs, and it is also one of the lead molecules which is being explored for many other conjugates and derivatives being used for cancer treatment, especially in chemotherapy. 4 of the drugs are already in the market, but there are more than a dozen derivatives and conjugates who are under different stages of clinical trials in FDA. So it is a molecule of good interest, knowing its increasing market demand, and at the same time, it's decreasing supply. It is coming from a rare species. It is an endangered species, mainly sourced from 2 plants across the world. Major source had been Camptotheca acuminata and Nothapodites pneumoniana. Nothapodites pneumoniana is native to India. It's also found in Southeast Asia, but it's majorly sourced in India through Nothapodites pneumoniana. It is there in Western Ghats. There has been a rapid decline in the population because of extensive uprooting. It is, I did mention in my earlier classes about this molecule that it has to be non-invasively sourced from the bark of the tree. But just because of the economics or commercial gains, it is completely uprooted. And there's a lot of smuggling of the raw material also going on. So there is a large decline in its wild population in the Western Ghats as well. Now, if you see the demand-supply gap, the annual global demand is somewhere around 3,000 kgs, wherein the supply is just 600 kgs. So it's almost 50%. If you see that this data I'm talking about few years back, maybe it has worsened. So there is the idea is to tell you that there is an increasing gap between the demand and supply for this molecule. So it becomes a worthy molecule to investigate and to develop a bioprocess, an alternative method of producing this molecule. So this is what I was mentioning, just to tell you about the importance of this molecule. I think Professor Nathalie, we have been now trying to work out some collaborations around this molecule in both the labs where camptothecin has been the molecule of interest. I think if I'm right, Nathalie, correct me if I'm wrong, that it has been recently declared as one of the essential molecules, essential medicines by WHO. Now, if you see the CAGR, it's rapidly increasing for this particular molecule. And knowing that India does have a particular source of this plant, and that too, it is an endangered plant now, it becomes imperative to try to find out alternative sources for this particular molecule. It's quite expensive, especially the derivatives which are used in chemotherapy. They're irinotecan and topotecan drugs. They are quite expensive, so which also makes the cancer treatment expensive. So this was the number of derivatives and conjugates of camptothecin which are under different stages of clinical trials. So camptothecin is topoisomerase 1 poison, and especially arrests the multiplication of the S phase. How is it produced? It is a monoterpene indole alkaloid, the same what Professor Nathalie was talking about, the MIAs, and it is produced using those same key secondary or specialized metabolism. The shikimate pathway, the mevalonic acid pathway, and the MAP pathway. The intermediates are coming from those pools. And major precursors is tryptamine and cycloganin coming from both these paths. They combine to give you strictocidine. Now, strictocidine is a very key molecule if you talk about MIAs. You must have, if you remember, even when she was talking about vincristine, vinblastine, strictocidine comes in the picture. Now, as you keep going downstream with different plant species, the end product keeps differing. Your vincristine, vinblastine, your camptothecin, catharanthine. So these are all, but the key precursor molecule is strictocidine. So what we did, I just spoke about that 10 years back we had seen, now more than 10 years, around 2010, there was a report that a nanogram yielding endophyte was isolated by UASB Bangalore and was submitted in the National Repository of IMTech Chandigarh, MTCC. So we thought, and the same reports we could find in literature that there were equal number of papers saying that they cannot be sustainable producers, but at the same time, many reports were coming where the culture conditions were enhancing or reviving the production. So as a bioprocess engineers, we thought that if really it is true that the minute you isolate it, the biosynthetic machinery is getting cryptic because of the in planta selection pressure or in planta stimuli which is missing, can process optimization, which means manipulation of the culture environment, can help the organism to revive. So with that hypothesis in mind, we picked up that nanogram yielding strain from the public repository, and we tried to explore all those strategies of productivity enhancement and yield enhancement to see if something can enhance it from nano to some fold enhancement. Just to develop a proof of concept, we were also trying to see whether it is a worthwhile investigating subject or not. So this was a master's thesis project. So we were trying to see if by manipulating or application of elicitation, let's say precursor addition, culture condition optimization, is that bringing any change in this particular production of camptothecin? Because if that brings in the change, what gets proven? The proof of concept that possibly the biosynthetic machinery is present in the organism, but it might be cryptic. So with that hypothesis, my first master student, she is in UTA, I think must have graduated also for PhD. Now, Aarti then wanted to explore with this particular endophyte. Now, even to quantitatively find out camptothecin from a nanogram yielding strain, it was not easy. So we had to develop a protocol, both at qualitative and quantitative level, to be able to identify the molecule at such lower concentrations. So then what we found, so there was I would say this was a serendipity. So she was exploring, we were trying to, because we knew that strictocidine is the key precursor molecule. So we thought, let's try to work out. So this was the hypothesis which we drew. We knew that the strictocidine has to form to produce camptothecin. And there were some reports in which horizontal gene transfer hypothesis was also being published between the endophytes and the host plant, saying that possibly that could be the reason why they can be potential producers because of these horizontal gene transfer happening. So we were thinking that possibly if that is true, and we know that strictocidine is the key precursor molecule, is the rate limiting step. What if we add strictocidine as a precursor and see if camptothecin production is enhanced in the culture? Our simple precursor addition strategy, which we use in plant cell technology, we started finding out if we could find strictocidine in the market. Now I heard when I went to France that they were producing strictocidine, but that time, 10 years back, there was no strictocidine in the market. So Sigma also could not give it to us. So then we thought this doesn't seem to be working. Then we figured out Vinca rosea, those who were working on vincristine, vinblastine, that group. It's ubiquitous here. It grows everywhere in the campus. So the student said it was a master's thesis project, completely exploratory. She said, ma'am, can I try the extract of Vinca rosea? And if cryptocidine is present in the extract, let's add the extract and see if something happens. And then we'll do LCMS to identify the molecule. I said, okay, you try. So then she came back after doing so. We never used to meet every day, but maybe bi-weekly, if I remember it right. So she came and she was so elated and happy, jumping with joy that, ma'am, 50-fold, 15-fold enhancement in camptothecin. I said, okay, cool down. Go back and do this experiment 10 times and then come back to me. So then she said, okay, ma'am. So then she went back. She did multiple times and she was all the more apprehensive than me. So she did multiple times that experiment. She was a perfectionist. So she did multiple times that experiment. She came back to me, ma'am, it is working. It is working. I said, okay, show me the data now. So I saw the data and I said, where is the control? So she said, oh, I forgot to use the control. So control was what? Because she was using an organic solvent extract, possibly to extract out cryptocidine. So then I said, did you grow alone in the organic solvent? How will you find that the culprit is not your small molecule, but your extract? So she said, okay. I said, you go back and do your control experiment and then come back to me. She did her control experiment and she came back again running to me after completing the experiments. Although a little sad, she said, ma'am, control is giving me better result. So, so then I said, I'm more happy to know that. You know why? Because to investigate a complex extract is much more difficult to take forward than knowing that a small molecule is doing a job. You have now in hand a single molecule which is now doing a job. Much easier to take the experiments forward. So I said, repeat it and then come back to me. And this time, do an experiment in graded concentration of that small molecule and come back to me doing it replicates. So this will give further an idea that it is indeed that small molecule and how is that small molecule. If it is in graded concentration also giving you proportionate increase in camptothecin, further proves your POC. So I said, she came back, she said, ma'am, up till 5% volume by volume. And that small molecule was ethanol. I said the culture is, and what about the biomass? Is the culture, which means the culture was tolerating 50 grams per liter of ethanol. I said, okay, you show me the biomass. I saw the biomass is also increasing. The camptothecin is also increasing. That was very, very interesting. So then the question came, what role is this molecule playing in the culture? So I said, okay, being a master's thesis now, some things are coming to close. Thesis submission has to happen. She has to move out. So then I said, okay, let's do those final round of experiments. You try to now investigate. As a last part of the thesis, it will be very nice to close the story by at least some indications on what role is this molecule playing, which has led to this enhancement possibly. So then she did some total protein estimations. She did some total NAD estimations. She did also investigate with increase in the graded concentrations, what is the optimum ethanol concentration up till which it is increasing continuously. So what she could find was from 0 to 10% volume by volume, she could find at 5% it was giving best results. Beyond that, possibly because of the toxicity, the viability loss, it was reducing. So this became an interesting proof of concept established now for us to become interested further in endophytes, that possibly it has something to do with the cryptic biosynthetic machinery, just because they are now in the exinic state. So then we laid this hypothesis that possibly this is what is happening. Ethanol might be acting as a stimulator to enhance, to give back that stimuli. Now, if you see, we are still investigating this, but in planta, the environment is not rich. Generally, when we are natural instinct, when you get the microbial strain and you want to grow it in the lab, you will give it a rich source of media, or complex source. So everybody is in the habit of using a PDA or a potato dextrose broth and try to grow it. These are all rich nutrient sources. So then we were thinking that is it something to do with the in-plantar atmosphere where the concentrations will be much less possibly, or some kind of stress? So then in this much time, we also realized that although the POC is established, it's worth investigating further, but may not be worth investigating such a low yielding microorganism, because although the molecule is of high value, but if the strain is such a low yielding strain and we are putting in so much time, effort, resources, and in next 5 years, although we got 15-fold enhancement, where did we end up in? Still in micrograms per liter titers. So is it really worth using the same organism, or it would be better to work with a much higher yielding organism and now investigate and implement all these similar strategies in a more rational manner to see if it can lead to comparable camptothecin titers, which one could think of in plant cell suspension cultures. That was the reference point. We were also in parallel working with Nothapodites pneumoniana plant cell cultures. So, or at least the yield should be comparable to what you can find in nature, which is around 0.4 microgram per gram. Sorry, milligrams per gram. So this is the same. I'll just skip this. So then we investigated the effect of different precursor additions, the effect of different elicitors also. And we could find in all these strategies that enhancement was happening. So where did we close the story? We closed the story by the finding that possibly the endophytes can be sustainable producers or can be potential producers of host plant associated metabolites, but the sustainability of the production would depend on the culture conditions and reviving that cryptic biosynthetic machinery which is present in these endophytes in the exenic state. Now then next PhD student came and I said we closed that story because it was not our strain, and first thing, and the second thing, it was a nanogram yielding strain. Now let's go back to nature and try to do some bioprospecting to find a much higher yielding strain, which can be a worth strain to implement all these strategies and see where do we land up. So with that thought in mind, Khwaja came in, and then Khwaja, this was in collaboration with UASB Bangalore. And because it's an endangered plant, you cannot go to wild and just start doing your R&D. So we did this work in collaboration with UASB, who were ecologists and who had this particular plant in their horticulture. And they helped us in the bioprospecting. Some 132 endophytes we isolated, and we marked from which explant are we isolating. And out of those 132 endophytes, 90 seemed to be producing in the exenic state, camptothecin. Remaining were not producing. Some were coming from the same explant, which were non-CPT producers, and some from the same explant were CPT produced. CPT is the short form hereafter I'll be using for camptothecin. So what did we did? We isolated, and then we did some screening to find out which can be the best strain to work with. And based on this quantitative and qualitative screening, qualitative was based on tandem mass spectrometry, which is MS-MS, LC, MS, and MS, fragmentation pattern also, so that you can be sure. And that time we had an Orbitrap, so the resolution was very high, up till 4 decimals. So, so as to ensure that, yes, indeed, what you're looking at is camptothecin. And then we also quantified it using HPLC. We ranked the list and we picked up the top 6 strain. Interestingly, the top 2, which were the best producing, the topmost was giving us closer to what you can find on an average in nature, 0.4 mg per gram, which means 400 micrograms per gram. And the second highest yielding was giving us nearly 200 micrograms per gram. So, but the question, we knew from the literature that it's about sustainable production. So we picked up these 2 strains and we did for 3 years followed up the culture, and we did some subculture studies. How is the culture behaving in suspension? So this was, when I say one subculture cycle, it doesn't mean directly from one suspension to another. The master cell bank, the working cell bank used to be prepared in as a slant, From that working cell bank, after 7 days, we used to get the spores. Those spores used to get inoculated in the suspension. And after 8 days, we used to extract out from suspension the camptothecin from the biomass, and then we used to investigate. So multiple generations were going through. So when we did that analysis, what we found was that the second highest yielding strain, as well reported in literature, we did find a dip in the production capacity of these two strains, both the two strains. First, very quickly, soon after the second and the third subculture, became very, very low. So they did show an asymptotic fall. The second highest was giving us again a fall, but it became again asymptotic, but where it ended up nearly around 50% of its initial value, which was now how much, around, I think, 200. So, correct. So I think both 400, let me check the yields. I made some mistake. Correct. So the top yielding was 426. The second top yielding was 403. And this 403, although was second ranked, was after 13, 14 subculture cycle analysis, it became stable around 200 microgram per gram. So, which is not bad to begin with. So then we thought, let's pick up the second highest yielding strain, which is demonstrating some steady now production after 14 subcultures, and investigate it further. So with that in mind, we brought it into suspension. We did bring it to the reactors to see again in batch, multiple generations would go by. What is the scenario? And we also, this is a part of the study where we verified whether there is a correlation which can be drawn between higher yielding explants vis-a-vis the higher yielding endophytes being isolated. We could find some correlation. Then we also went on to test the hypothesis that with how is it that the camptothecin producing endophytes, will they have more tolerance towards camptothecin than the non-camptothecin producing endophytes, which were being isolated from the same explant. So we picked up 3 of camptothecin producers, 3 non-camptothecin producers as pairs from the same explant where they were isolated. And we saw that those which were showing camptothecin production capability were having much higher tolerance towards camptothecin is a cytotoxic molecule, were showing much higher tolerance towards camptothecin than non-camptothecin producers. So this is what, although expensive, but this can become a very good screening methodology for if you are wanting to bioprospect a higher yielding endophyte. Okay, so this was that stability analysis study. If you see the highest yielding endophyte, very quickly up till 4th generation, it has gone to very low amounts. So this became a promising one, the second highest yielding. Now the idea was to further invest. So we did all TLC, H1 NMR, and tandem mass spectrometry, HPLC, to be sure that indeed it was camptothecin. Now to further investigate that this camptothecin, there were reports in literature that endophytes demonstrate in exinic state that they are producing camptothecin, maybe because they are storehouses of the host plant associated metabolite. So when the host plant is producing it, maybe because they are residing in the plant, they become storehouses for that metabolite. So we wanted to be sure whether it is coming from the metabolism of the organism or not. So what we did was, we gave total labeled, completely labeled glucose to a starved culture, and we did the entire suspension culture, and we extracted the biomass. And because it was an intracellular metabolite, we extracted camptothecin, and we wanted to investigate if the label is found in the camptothecin. And that is what we found. Camptothecin is a C20 molecule. So we could identify from C1 to C20, although C18 was the most relatively abundant, but you could identify the labels present in the extracted camptothecin, which proved that indeed it is coming from the cell metabolism with all our controls in place. And then we also did some comparative analysis with camptothecin standard, whether it is showing cytotoxic impact or not, the extracted camptothecin, although it was crude extract. But we wanted to do a comparative analysis. As expected from camptothecin standard, we could see that it was sensitive towards the lung and the oral cancer cell lines, which the camptothecin is known for treatment. We did other also, this was work done in collaborations, just to a comparative analysis with the standard camptothecin, that the camptothecin which has been extracted from the organism, how is it cytotoxic or not? How is it behaving against cancer cells? So all that was falling in place, and we were able to see that yes, it seems the organism indeed is producing camptothecin, first thing, and the camptothecin which we have extracted is the camptothecin which we are looking for. Then we started implementing all these optimization strategies, which you already know, and everything was showing us improved production of camptothecin. Where do we stand as of now? Now, because the culture is being stored in the as a master cell bank, indeed it is showing, because we are not keeping it under the stimuli, it is indeed showing continuous decrease. Now the point is, we are working on the hypothesis that because we found that adaptive, they had shown good tolerance towards camptothecin. Now we are exploring adaptive laboratory evolution to see if the tolerance towards the camptothecin can also give us a mutant which is much more revived from its initial cryptic state. That is one. The second is, we are also trying to test the hypothesis of horizontal gene transfer from the host plant. If indeed streptozidine, assuming, is the rate limiting step. So we are trying to explore if the host plant enzyme expression in this particular organism can lead to some changes in the camptothecin, if horizontal gene transfer theory is true. So that's also we are exploring. So a number of projects which are going on. We also did whole genome sequence. We did some transcriptome analysis to identify how is the organism producing this molecule, camptothecin. So although some of the steps are still unknown in plant, but at least we were able to identify putative gene clusters. This was again a collaborative work with a German lab, a putative gene cluster which seemed to have the upstream intermediates present in the organism. But it is all work in progress and very, very exploratory and very interesting. So let's see where we head. That's it. Thank you. So that is it. And this is the projects which are going on. Yeah. So this brings to an end of all the lectures in the course. I hope you had a good experience in the course and you could gain, and at least your interest in the area of phytochemicals could get ignited to take it forward in whichever way you would like to. And feel free to connect with us in future as well for any kind of guidance you would be requiring. And tomorrow anyways we meet, then I'll be giving my last word in this particular session. Okay. All the best.